All right, hello, I'm Nick Nedzesky, and this year I conducted my research experiment on the effects of glycerol on biofilm production. So I am in 11th grade, and this is my third year of laboratory research experience working in the Seneca Valley Center for Biotechnology Research. That is where I conducted this research experiment. It is a lab in the intermediate high school, and local universities donate supplies and materials for students like me to be able to work and conduct research experiments. So I'm very excited to tell you about the research that I did in this lab this year. So what exactly are biofilms? Well, that's a great question. Biofilms are communities of bacteria encased in an extracellular matrix composed of exopolysaccharides, which are sugars, proteins, and extracellular DNA. So I made this little computer graphic here to show exactly what a biofilm is. So when we have some bacteria and they're just floating in their environment here and they're swimming around looking for a surface, we call that planktonic. So these bacteria are planktonic bacteria and they're looking for somewhere to adhere to. And when they find that surface, they will attach and they'll start to replicate. And as they do this, they will be producing and secreting this biofilm. And this biofilm allows them to have protection against antibiotics, against the immune system, and against any other natural predator in their environment. So this biofilm is very helpful to these bacteria, but can be very harmful if they are a part of an infection in a human body. So why did I use glycerol in this experiment? Well, glycerol is meant to serve as an osmotic stress factor in this experiment. So, and keep in mind, this diagram is not drawn to scale at all. It is a very oversimplified representation of a bacterial cell membrane. So this is just one bacteria, and there's no cell wall pictured whatsoever. So it's very oversimplified. Glycerol is not huge like that. It's much smaller, but it's supposed to be a good visualization of how glycerol will diffuse into bacterial cell membranes. And I hypothesized that that would cause a great deal of osmotic stress for these bacteria. So as diffusion and osmosis is occurring, these bacteria will be put into a lot of osmotic stress, and that will induce the WISP pathway, or the WSP pathway, which is the main biofilm producing pathway that these bacteria use. And the WISP pathway produces a secondary signaling molecule known as cyclic di-GMP, which is the molecule that the bacteria use to signal their biofilm production. So they will produce a lot of cyclic di-GMP in order to signal that they are ready to produce a lot of biofilm. And vice versa, they'll produce less cyclic di-GMP when they're ready to stop producing as much biofilm. So looking at the big picture here, I believe that glycerol and the osmotic stress that it will induce the WISP pathway and allow for increased biofilm production. And I also believe that this glycerol will act as a selection pressure for these bacteria, so that those bacteria that aren't better fit for biofilm production will die off. They won't have as much biofilm to protect them, and thus I will select for only the best fit bacteria that will produce the most biofilm, and I will have a lot of high biofilm producing mutants. So as I'd said, I expect, as a part of my hypothesis, that glycerol will induce biofilm production, and that it will also select for high biofilm producing mutants, which will provide these bacteria with more protection. As you can see here, I tested this hypothesis with 2 trials using 5 culture tubes, using 0%, 2.5%, 5%, 7.5%, and 10% glycerol media. And I cultivated Pseudomonas fluorescens, which is a biofilm-producing strain of bacteria, in 2 milliliters of each of these media. And I incubated these tubes in an orbital shaker for a week at 28 degrees Celsius and 100 revolutions per minute. And after a week of experimentation, of incubation, and 2 tube transfers, I plated a sample from each of the tubes, in order to qualitatively analyze their colony morphological differences. So also, I stained and measured data with a dual beam spectrophotometer in order to be able to obtain quantitative data as to the effects of glycerol on biofilm production. As you can see here, I have a table of the media recipes that I had to develop myself in order to carry out this experiment. I had to fluctuate the amounts of glycerol and water in order to achieve the concentrations that I wanted, and I kept essential nutrients constant in order to ensure that only the change in glycerol is affecting how these bacteria are growing. And as I mentioned earlier, there were 2 tube transfers done, mainly to replenish the media as the bacteria consume it, and additionally to allow for the continuation of these generations of bacteria as they're growing and mutating on these tubes. So as you'll see here, I have an ancestral control plate where I have my bacteria growing that weren't grown in the presence of glycerol. And I'll circle on this plate just some smooth and round colonies that are notable examples of just the standard wild phenotype that you would expect in Pseudomonas fluorescens. It's smooth, it's round, and you'll notice that there are some small amounts of wrinkly colony mutants known as wrinkly spreader mutants, and you'll see them all throughout the plate. They have a little bit of a thicker border there and a wrinkly edge to them. So that's why they're known as wrinkly colony mutants. And in this control plate, there's not too many of them. It's actually mostly smooth and round colonies. But later on, we'll see in the 10% second trial plate and in every successive plate after the 0% and 2.5% plate, we will see increasing concentrations of wrinkly colony mutants. And you'll see those circled all throughout this picture. There's a lot more wrinkly colony mutants, significantly more in each successive plate. And there's a larger picture of that down below. And in order to get quantitative data, as I had mentioned earlier, I developed and experimented with this staining procedure. So I used 0.1% crystal violet stain in order to stain each of the tubes. I washed them using water, and I stored them in ethanol so that they could be processed using the dual beam spectrophotometer. Now, a spectrophotometer works by shining light through samples and then measuring how much of that light was absorbed and how much was able to get passed through. So I wanted to look at absorbance in my case because that is a really good marker for how much biofilm is being produced, because the more biofilm there is, the more absorbance that I will read because there'll be more stain present in the sample. So here are my results. I wanted to show raw data here just for a second because I wanted to show some of the errors that I, as a researcher, had to overcome. You'll notice that there's data, a lot of data actually, highlighted in red, and those are omitted outliers, okay? Those are data points that I knew were not correct because those tubes had undergone a destaining phenomena. So as I had added water and washed some of the tubes, some of them ended up losing their stain. And that was a big issue, a large source of error in this experiment. And so in my final statistical analysis, I omitted the tubes where I could visibly see unstained chunks of biofilm floating in the media, and it was kind of a big mess. So I omitted those, but I still wanted to include them to show some of the hardships that I had to overcome in this experiment. Additionally, I was not able to collect any data for the second trial 10% tube because it had actually gotten destroyed in a centrifuge. I had tried to load it into a centrifuge to try to see whether or not I could concentrate the biofilm at the bottom, but from what I heard from my lab director, that centrifuge had not been used in a while, and I had done everything properly. I loaded it in, I had a counterbalance, I had everything properly in there, but the centrifuge was faulty and the tube was just torn up inside of it. So I suppose that's just what happens in a lab. But I did get some good data here. I did get some salvageable, really good data, actually. If you'll look here, I'm using the Pearson correlation coefficient in order to determine exactly how positively correlated this data is. So the closer these numbers are to 1, the more positively correlated they are. And as you see, these values are very close to 1. And the p-values associated with them are very low, meaning that this is very significant data and very significant correlations. Although keep in mind, there are some inconsistencies. There are some r-squared values that are a little low, not the best linear fit there. So I will definitely need to run more trials and collect more data, first of all, to make up for some of the data that was lost in the destaining effects that I had observed, and then also to collect more data to more concretely support my hypothesis. But overall, I would have to say that there was significant positive correlation between glycerol concentration and absorbance, which, as I said earlier, is used as a measure of biofilm production. And as we saw in those photographs, there were visibly larger amounts of wrinkly mutants present in each successive plate. So I would say that this experiment was a very great success, and it suggests that glycerol could in fact be an inducer of the WISP pathway and a selection factor for high biofilm producing mutants. As I mentioned earlier, there were a lot of limitations, a lot of error in this experiment. I'll definitely need to tweak that staining procedure in order to be able to collect more accurate data. And I'll also need to run my plate samples as I had shown earlier. I'll need to dilute those a little bit better so that the colonies are more countable, so I can get tangible numbers and percentages of wrinkly colony mutations on each of the plates. I mentioned earlier, I think I mentioned at least, that COVID-19 restrictions limited the experiment, and I actually wanted to show a calendar I had developed where I had to just kind of chart out my schedule for how and what days I was going to be doing what things and how. And I ended up on some days that the school was closed and the lab was closed, I ended up having to do a Teams call on Microsoft Teams with the assistant lab director, and he performed like some of the parts of the experiment while I directed live over Teams. So we definitely had to brainstorm some unique solutions to actually being able to get into the lab and work. Although for my entire second trial, I was able to be there in person. And as I mentioned, I'll need to collect more data. And another thing I thought was that I should probably sequence the mutations that I observed in order to confirm that these mutations are WISP pathway related. So that's definitely something I can do in future trials or as a future experiment to see exactly how I could select for certain kinds of mutations and to see exactly what I'm looking at here in those final plates. So why should you care? What is significant about this research? Well, biofilm-producing bacteria are responsible for approximately 65 to 80% of all human bacterial infections. And as I mentioned earlier, oftentimes these bacteria are antibiotic-resistant and resistant against the immune system, making infections with these bacteria very harmful, oftentimes very fatal. So there is high demand for research regarding biofilm formation and the treatment of these infections. So these are just some of my sources that I had used in my preliminary research before I began experimentation. It's always good practice to show some of those. And that is all for this experiment. So thank you very much for listening and have a nice day.